Hello, welcome to Chapter 7, the Central Nervous. Hello, welcome to Chapter 7, the Central Nervous System. So the Central Nervous System, CNS, is the brain and spinal cord. And in general, we say it integrates, coordinates, processes. And outside of the central nervous is the PNS, the peripheral nervous system. And that would consist of nerves bringing sensory information in, that is the afferent nerves, and the nerves carrying information out, that would be the motor nerves. So the sensory division of the peripheral nervous system, so remember we're outside the brain and spinal cord, we're in all those neurons that are coming out of the spinal cord. They bring information into the CNS from receptors, so sensory receptors. And these sensory receptors will bring us all kinds of information: light touch, deep touch, pressure, pain. We saw that in the skin. And it will also bring in visceral information from our organs. Viscera is organ. And it will bring in information from our head that provides special senses such as smell, taste, vision, hearing, and balance. So the sensory division is bringing information in. The motor division is taking actions out. The motor division carries motor commands from the brain and spinal cord, the CNS, to target organs called effectors. And we don't have a lot of effectors. Think muscles and glands. Muscles and glands. Somatic is body, so the somatic nervous system takes information to our skeletal muscles. We have control over that. Autonomic, think automatic. We don't have control. Automatic. The autonomic nervous system sends information, motor information, out to our smooth muscle, our cardiac muscle, and our glands. You can skip the adipose tissue. Picture is good. The purple is your central nervous system where there's processing. The yellow is your peripheral nervous system, everything outside your brain and spinal cord. So we start at our receptors. We have 3 basic types of receptors: somatic, special sensory, and visceral. Remember, somatic is in the body. Think of what's in the skin: pain, pressure, temperature, light touch, deep touch, that sort of thing. Visceral would be our organs. And since our organs full? Are they stretching? And then special senses are those senses in the head. So the sensory division is afferent. Incoming information first. A comes before E. So the incoming is the A word, afferent. It's going to be processed. Information is going to be received, and an action is decided upon. And that action can only take place by the motor or efferent division, and that includes the voluntary and the involuntary. The voluntary is the somatic nervous system, our skeletal muscles, and the involuntary is autonomic nervous system, smooth and cardiac muscle, and our glands. We have no control over our glands. You need to know the features of an axon. Not all the stuff here, but we know cell body, you know cytoskeleton, the nucleus, nucleolus. The only difference here is we have these extensions on the cell body called dendrites, and information comes in the dendrites or the cell body, and information that is a nerve impulse goes out the axon. So information is in a one-way, flows down a one-way street out the axon. And this axon can branch and have what are called collaterals, and then the axon is going to have axon terminals, and at the end of the axon terminal is a knob. Now the synapse is this connection between this neuron, the blue one, and the second neuron, the purple one. We saw this in the muscle chapter with the neuromuscular junction, where instead of synapsing with a second neuron, the motor neuron synapsed with a muscle cell, but everything else is almost identical. And here's a cool statue of a neuron in San Francisco that I saw. Now, we have names for different kinds of neurons, but you have to understand how to call it that name. And to do that, you're going to look at the cell body. So at this one, look at this cell body on the left here, and coming off the cell body are two processes. Therefore, this is called bipolar. Now, this is found in some sensory organs. We see it, say, for example, in the eyes. Here we look at this cell body. Really, there's only one process coming off, but it splits immediately. But there's only one process, so it is unipolar. And most of our sensory neurons bringing information into the body are unipolar. This is the most common in the brain and spinal cord. This is your typical axon neuron that we learn about. We learn all the different parts of this neuron. And there are many processes coming off the cell body, so it is multipolar. Stem cells do exist in the nervous system, but not all over. Therefore, most of our neurons will not divide or be replaced when damaged. But we do see some multiplication of these cells in our olfactory region, our patch for smell in our nasal cavity. We see it in the retina, and also the hippocampus. Now, the hippocampus is a part of the brain. We don't really spend much time talking about it, but it is involved in short-term memory storage. And they studied the hippocampus of people who ate a lot of junk food, and they had smaller hippocampus. What part of the neuron transmits electrical signals away from the cell body? That would be the axon. What type of neuron has the cell body located off the side of the axon? Off the side, that would be like one connecting process. That would be unipolar. What part of a neuron carries an action potential? That would be the axon. So there are other cells in the nervous system besides neurons. And those cells are called neuroglia, they're called glia, they're called glial cells, and half of the cell volume are neuroglia. They are support cells. You do not need to memorize each type of support cell and what it does, but we need to understand what some of these cells do. You won't be tested on these guys. Well, number one, the ependymal cell. We have cavities right in the middle of our brain, and these cavities make our cerebrospinal fluid because they're lined with these ependymal cells. So the cerebrospinal fluid is produced in the spinal cord and brain by these ependymal cells, and it circulates around. We also have some phagocytes that can destroy any infection or any problem that occurs, and those are very small cells called microglia. They are phagocytes. We have cells that we'll talk about blood-brain barrier in a second, but these astrocytes are the go-between between the neuron and the capillary. So the neuron is flowing by, the astrocyte will grab the nutrients and oxygen from the capillary and supply it to the neuron. Neurons can be quite large. Think about the size of a neuron in a giraffe that has to travel from his spinal cord down to the bottom of the foot. So they can be 6 feet long or longer. They need some support. Astrocytes do form scar tissue so that our neurons can't regenerate, but they're important up in the brain because they prevent things in the blood from getting into the brain. So those things in the blood might be wastes, but they also might be drugs we're trying to get to the brain. So when they do brain drug research, they have to get past that blood-brain barrier. And then oligodendrocytes produce a fatty coating around the axon called myelin. The whole purpose of this, I know you think fat insulation, but no. The purpose of the myelin is to speed up the impulses. The myelin sheath, where it's myelinated, is called an internode, and that would be like a part of a cell, and then there'd be a gap, and then another myelin sheath, and the gap is called the node. Where you've probably heard the terms gray matter and white matter. White matter is regions that have myelin. So where there's white matter, you know there must be speed of transmission. Speed is essential. So white matter is for speed, and it's myelinated axons. Gray matter, on the other hand, could be unmyelinated axons, no speed there, or the cell bodies. They are also gray matter. So here's a picture of all the different types of neuroglia. Our first one was the ependymal cell. So here is a ventricle in the brain, or the central canal of the spinal cord. And these ependymal cells make cerebrospinal fluid. We have oligodendrocytes, where here's this weird guy here. This cell is allowing its plasma membrane to wrap around the axon, forming the myelin sheath. And it can wrap around a bunch of them. It's kind of wrapping it like a burrito here. Look at all the layers. The astrocyte is the middleman between the capillary and the neuron. So the neuron is blue. This capillary, remember, is just simple squamous epithelium. So the nutrients from the capillary diffuse through the astrocyte to get to the neuron. And then finally the microglia, tiny little cells here that act as phagocytes. Now, we've just been talking about the central nervous system. There are different cells in the peripheral nervous system, so you didn't have to know the names of those in the central nervous system, but you do have to know the name of the Schwann cell in the PNS, and the Schwann cell is what makes the myelin sheath. It wraps around just like the oligodendrocyte, but it's just a single cell supports a piece of an axon. And then there's some support cells that kind of act like astrocytes. They're called satellite cells. In the PNS, clusters of cell bodies are called ganglia. So here we are, we're in the PNS. So when an individual cell wraps around here, that's a Schwann cell. So 1, 2, 3, 4, 5, 6 Schwann cells, and they've cut this one in half. We're in the PNS, and it forms the myelin sheath for speed of transmission. And then we also have satellite cells to assist in, like the astrocytes, maybe a little bit of nutrition, just some support cells.